Welcome to the 2019 Community Education Forum. I'm Ariel Walker, co-founder of the Huntington Society of Canada. When my husband Ralph and I created the society over 45 years ago, right here at our kitchen table in Cambridge, Ontario, we never imagined our community would grow like it has. We envisioned the Huntington Society could offer support to families affected with Huntington's and fund promising research. The resources that the Huntington Society offers families and the research being undertaken today is everything that we had hoped for and more. Back then, the HD gene hadn't been identified yet, and here we are today with clinical trials addressing the root cause of Huntington's disease. I'm hopeful for the future because of our dedicated researchers and know we will continue to learn more about Huntington's as we move forward until a treatment is available. Today, we have the privilege of hearing from two researchers, both longtime friends of the Huntington Society, who will inform us about the progress in Huntington's research and provide a glimpse of where we are headed. I hope you're just as excited about all this as I am.
Hello, this is the voice of Bryn Del Mayo, Vice Chair of the Huntington Society of Canada. Thank you for watching. It is certainly a hopeful time for the HD community right now, which is why we wanted to bring you leading HD experts to provide updates in research and clinical trials. Today we're going to hear from two researchers about current trends in Huntington disease research. First, we have Dr. Michael Hayden. Dr. Hayden is a Killam Professor of Medical Genetics at the University of British Columbia and a Canada Research Chair in Human Genetics and Molecular Medicine. He is a senior scientist at the Centre for Molecular Medicine and Therapeutics, which he founded. Dr. Hayden was the President of Global Research and Development and Chief Scientific Officer at Teva from 2012 through 2017. During this time, approximately 35 new products were approved in major markets, with many for diseases of the central nervous system, and led to the approval of Austedo for chorea and HD, the second drug ever to be approved for use in Huntington disease. In 2015, Teva Research and Development was recognized as one of the 10 most exciting innovators in pharma by Idea Pharma, and in 2017, Teva R&D ranked top of the industry for CNS development and clinical trial success by Pharma Intelligence. Dr. Hayden has also been awarded the Order of Canada in 2011 and the Order of British Columbia in 2010. Dr. Hayden is committed to empowering others. In addition to mentoring over 100 graduate students and postdocs, he is also a TED mentor. In his presentation, Dr. Hayden discusses the different components of each phase of a research trial and how there are many HD trials being conducted with different approaches, not just one. He emphasizes the importance of managing our expectations in this exciting time of research, but also encourages us to have realistic hope for the future of HD. Without further ado, here is Dr. Michael Hayden.
Well, I'm so delighted to have this opportunity to talk to members of the community of Huntington disease in Canada. And I'm really thrilled to just share with you a little of my thoughts about the stage we're at, what we can expect, and also what we can expect relative to managing our own expectations. So these are indeed wonderfully exciting times for the HD community. For the first time, we have numerous drug trials from many different companies in process, in progress, or being planned. And these are companies from all over the world, companies in Europe, companies in the United States, and for the first time we're seeing an amazing interest in the Huntington disease community and also companies willing to sponsor trials throughout the world. And I wanted to just go through with you a little bit about how trials work, how we think about clinical trials, and what you can expect as these trials progress through different stages. Generally, before a trial occurs, there's a lot of preclinical data. Preclinical means it hasn't yet been in humans. These are results of experiments in test tubes or in animals, which give validation or give support for the use of this drug in clinical trials. With that information and information about animal toxicology and toxicity, as well as information about the clinical pharmacology or the half-life of the drug and what happens, one then develops a model for what could happen in human studies. And in that process, with all regulatory agencies, you really get permission to have a first-in-human study. This first-in-human study is heralded by an IND that you file to the regulatory authorities, where you outline, the IND is often thousands of pages long, and it outlines the protocol, it outlines the number of sites, it outlines the outcomes, both the primary and secondary outcome that you're desiring, and it outlines all aspects of the background of this drug, including all aspects of the preclinical safety, pharmacology, and efficacy in different models, both in test tubes as well as in animal and in vivo studies. When you file your IND with the FDA, this takes 30 days, and the FDA often has some comments to make, but then after 30 days, they usually, but not always, allow you to go into human clinical trials. The first phase of human clinical trials is generally called phase 1. The time taken to get from the time you have a concept through all these preclinical studies till the time you file an IND can be anywhere between 3 to 5 years. So it's a long time that it takes to go from a concept just to get the drug into humans. Of course, you have to also have the manufacturing sorted out, have the drug produced in the right quantity, and it has to be appropriately done according to regulatory standards. And then you're ready to apply for the first time, and the first phase of this is a small trial that is called a phase 1 trial. It's really a safety trial. Occasionally, you try and include some things in that trial that will give you hints of target engagement or efficacy, but generally this is a safety trial, and that's the purpose of phase 1. So when you do phase 1, there you have various monitoring for safety. It's not usually a long trial. You can start off with a single ascending dose of what we call a SAD, where you just give one patient and then you give one dose, then you go to a higher dose and a higher dose. And then you can also start with a multiple ascending dose, which are all part of phase 1 safety approaches with some hints of efficacy depending on endpoints. In Huntington disease, it's very hard to look for endpoints too early, but you can, for example, as happened in the Ionis trial, look to have knockdown of mutant Huntington, and that can be an endpoint. It's not an efficacy endpoint, but is an endpoint potentially of target engagement. The likelihood of a drug getting from phase 1 to being available for patients on the market is really in the likelihood of 5 to 10%. Most drug trials fail, and only a small proportion get through. But if you have a successful phase 1 where it's safe, and essentially the FDA will allow you to go to a proof of concept trial, which is a phase 2 trial. The phase 2 trial is a smaller trial, but now you have some efficacy endpoints. So in Huntington disease, you may have total motor score, or you may have total functional capacity, but you would do a trial that looks for some endpoints. Generally, they're primary endpoints, secondary endpoints. The FDA would agree with that. Again, you'd file a protocol to the FDA, get permission to start, and often the phase 2 trial may happen in many sites, usually may involve up to 60 to 100 to 150 patients. You always would have a placebo and a treatment arm. And the purpose of phase 2 studies is to determine, firstly, whether you have a signal for efficacy. It may not reach statistical significance, but it is important to look for a signal for efficacy and also to determine the dose. So often in phase 2, you will go with more than one dose so that you look at the optimal dose for safety. Now, when you are in phase 2, the likelihood of that drug getting to the market or getting to patients so that you can get this as a script in your pharmacy is only in the range of around 35%. So about 1/3. 1/3 of them will reach an efficacy endpoint that looks okay, that allows you to plan now for a detailed phase 3 study. Now, a phase 3 study, when you after your phase 2 study, you'd have the end of phase 2 meeting with the FDA with what you've learned. You've learned that this is, for example, safe. Now you have a dose. Now you have some endpoints that you can do in a clinical trial. And then you go into phase 3. Phase 3 study, once you're in phase 3, the likelihood of success is anywhere between 60 and 70%. In other words, around 30% of phase 3 studies, 30 to 35%, will fail because they won't reach their endpoint. Generally, the FDA likes to have 2 phase 3 studies that really validate this for approval for being listed on the market. And these phase 3 studies are much larger, often involving many sites around the world, hundreds of patients, and they often take a long time. So in Huntington disease, if you were just looking for a movement disorder and looking to curtail, have impact on the movement, you could see that relatively quickly, maybe in about 6 months. But even for a 6-month study, the timing of the study is determined by the time the last patient was enrolled. So if it takes you 6 months or 8 months to enroll the number of patients you want, then the study would last another 6 months because the last patient has to be followed for 6 months. If the study is larger and you have a year study, so for example, if you want to look at total functional capacity as an index, the minimum you can look at is about 1 year, which means that the last patient that you enroll has to be followed for a year. So generally, when you look at that, accumulation and the ability to enroll patients will take you up to maybe enroll and then finish the study with a year. It's about 2 years. So by the time you get results there, it's at least 2.5 and could be even as high as 3 years, depending on the amount of time that you need to follow the progression. Now in Huntington disease, which is an orphan disease, the orphan disease designation for all these patients allows the FDA and other regulatory agencies to be a bit more permissive. Generally, they'll allow you only to do 1 phase 3 study. And then also, after that, so that shortens the time. And then also, certainly in the other approach to this, being an orphan drug designation, they may occasionally, if you have some indices for target engagement, allow you to go from phase 1 to phase 3 without showing the efficacy in phase 2. And so, for example, in the Roche trial that is starting sometime in 2019, there were some exciting results. The first was that the drug was safe. This was an ability to knock down mutant Huntington and wild type, as it didn't distinguish between both mutant and wild type. So it knocked this down, and this was seen in a dose relationship in the CSF looking at this. The efficacy is really not determined, but because there was some evidence for target engagement by knocking down mutant Huntington, the FDA agreed to allow Roche to go to a well-powered, very significant duration study for immediately to phase 3. But when you look at a phase 3 study, even starting in 2019, you're not going to get results before 2022, 2023, so it's a long time. Also, for that particular study, one has to also mention that as exciting as it is, there's no proof that the drug works. There's no proof. You do get a lowering of mutant Huntington, but we actually don't know in the CSF which is the fluid around the brain, where the mutant Huntington comes from. It's likely only to come from the superficial parts of the brain. And Huntington disease involves the superficial parts of the brain very diffusely, but also involves very deeply deep parts of the brain which are less exposed to that particular knockdown technology. So this is a very important experiment. It's a drug trial, but there is no proof at this time of efficacy, and this will have to be monitored. So it's important that people understand that this is just one approach. There are other approaches going on too. Another company is also looking at selective knockdown of mutant Huntington, just focusing on the mutant protein and not on the wild type. And that's another approach, using intrathecal or injection into the spinal cord, giving once a month for an extended period. So these are all trials that are going on. There's some other trials that are looking at small molecule approaches, taking tablets by mouth. But in fact, it's hard to know where the breakthrough will come. All of these have good rationale, have good preclinical data, have shown to be relatively safe in humans, and now are embarking on more efficacy trials in phase 2 and phase 3. Of course, these trials represent a unique partnership between the investigators, the companies, and the community, people like you around the world who are volunteering. And we are inextricably bound together in an effort to overcome the terrible ravages of this disease for you and your children, and hopefully for your grandchildren. You know, I think back to when I started working in Huntington disease in South Africa in the mid-70s, when I saw my first patients. And I think of the days meeting Marjorie Guthrie in the late 70s, where really it was hopeless and helpless, and there was tremendous darkness. There was no hope on the horizon. We didn't understand the cause. We knew it was genetic, but the gene had not been defined. And we compared this to today. There's been amazing progress, amazing collaboration and cooperation. The Huntington Society of Canada, as well as other societies around the world, have played such an amazing part as partners in advancing the field. And as I look to the future, after a long history in this field, I couldn't be more excited. At the same time, I'm realistic about the expectations. There's no proof that any of these drugs will work. Hopefully one or two of them will work. And just as we see for cancer, it is likely that there won't just be a single drug. It's likely that there's going to be a need for combination therapy. We are understanding more about symptomatic therapy. The second drug for Huntington's was just approved a few years ago, which overcomes some of the chorea. This is Austedo, which results in decreases the dopamine, so decreases chorea. So we've had 2 drugs now approved in the United States for Huntington disease, but none of them have any impact on the progression of the disease. And we're hoping now with these new approaches that there may be a chance to truly alter and slow down the course. But the odds are significant. From the time you have a concept to the time you get a drug to market, it can be 12 to 15 years. From the time you're in phase 1 till the time you get to the market, generally at least 5 to 7 years. So this is a long journey we're all on together. We're fellow travelers, carrying the load together, working together as a team in an effort to bring hope and promise to families with Huntington disease all over the world. It's a great voyage that we're on, and at the moment it's filled with numerous opportunities for success, and I think we have a real chance in the medium-term future, not short-term, to change the course and the hopes and aspirations of families with Huntington disease around the world. So the purpose of me talking to you today is really to give you some perspective, to manage your expectations, but also to give you hope, realistic hope, that this actually will lead to significant improvement in what is needed. Also, of course, it's very important that there's not just one approach. There are many approaches from many companies, and these companies now are seeing the value for the first time. Again, very encouraging that companies are investing in Huntington disease. The reason they're doing that is, firstly, Huntington disease is devastating. There's no real treatment to change the course of the illness. And so it's hope and also an opportunity to have real impact on patients' lives. But also, What you learn for Huntington disease, for example, if you're treating some of the secondary biochemical pathways, mitochondrial dysfunction, cellular dysfunction, intraneuronal calcium disturbance, and a whole host of other cellular abnormalities, these are exactly the abnormalities that also occur in other neurodegenerative diseases, from Parkinson's to ALS to Alzheimer's and to a host of other childhood neurodevelopmental disorders. So there's also a hope that if we are able to, and for Huntington disease, we know who's going to get it, by inheriting the mutation, depending on the CAG length, you are destined to be ill, but not yet ill. So we're able to define a group of patients, and then we're able to enroll them in a trial in the appropriate way. The long-term vision here is just as statins were used essentially to prevent coronary disease, is that one day we may have a medication that you can take when the moment you know that you carry the mutation, and that one day you'll be able to take something that will slow down the onset of your illness and delay the onset. That would be the dream. We're not there yet. We're some far away from it because you first need to show that this is going to be useful in patients who are diagnosed with the illness. But that's the long-term vision, the long-term hope, the long-term aspiration. And together, we can make tremendous progress. So I thank you for your time. I thank you for the opportunity to share some thoughts with you. These are personal thoughts. There may be some investigators who don't agree with everything I've said, and that's fine too. This is a community that welcomes debate and discussion in an effort to bring the best treatments, the best hopes, and the best possibilities to families with Huntington disease around the world. So thank you for your attention and your patience, and to all of us, safe travels as we go down this journey. Thank you.
Thank you, Dr. Hayden. Next, we have Dr. Ed Wild. Dr. Wild is a consultant neurologist at the National Hospital for Neurology and Neurosurgery and Associate Director of the UCL Huntington Disease Center, where his research team is focused on accelerating drug development through discovering new biomarkers for Huntington disease. He leads an MRC-supported longitudinal biomarker study called HD-CSF and is a global chief investigator of the HD Clarity study of cerebrospinal fluid in Huntington disease. He led the development of the first measurement platform to quantify the mutant Huntington protein, the protein which causes Huntington disease, in cerebrospinal fluid, and the discovery of neurofilament-like protein as a first blood biomarker to predict onset of Huntington disease. Dr. Wilds is a senior advisor and investigator in the Ionis Roche program to lower the production of mutant Huntington. He won the Huntington Society of Canada Community Leadership Award in 2012, the Huntington Disease Society of America Research of the Year Award in 2014, and Huntington Study Group Insight of the Year Awards in 2015 and 2017. Dr. Wild also co-founded HD Buzz, the leading source of plain language research news for the global HD community. Today he will discuss new treatments for HD, where we are now, and what's coming next. Ladies and gentlemen, Dr. Ed Wild.
Hi everybody, my name's Ed Wild. I'm a principal investigator at UCL Huntington's Disease Center in London. I'm a consultant neurologist at the National Hospital for Neurology and Neurosurgery. I am the co-founder of HD Buzz, which is your source of reliable information about Huntington's disease research. And I'm a long-standing best friend for life of the Huntington Society of Canada. So I'm really honored and delighted to be able to talk to you today about research in Huntington's disease and give you a big research update from things that have been happening recently. I'm focusing my update on treatments. Sometimes I would talk about things that have been done to mice to help us understand Huntington's disease, but my talk today is really all about things that are either being done in human patients now or will be expected to reach human patients very soon. Because really we're in a new era now of Huntington's disease therapeutic research, where all of the understanding that's been made since the gene was discovered in 1993 is finally making its way through into targeted trials of drug treatments that should, if we are lucky, make a real difference to the lives of Huntington's disease patients and their families, and hopefully together we can create a new future in which Huntington's disease is much less of a problem. So where are we now and what's next? First, we need to talk very briefly about the cause of Huntington's disease. What causes it? Now, if this was a talk about Alzheimer's disease, I wouldn't be able to make any sense at this point. I'd be able to show you photographs of people's brains and the abnormal buildup of proteins, but we'd really have no idea why those proteins build up or where they're coming from. In Huntington's disease, because we know what gene and what change to that gene causes the disease, we've had this really strong foundation from which to understand how the disease harms the brain, and that has formed the basis for all of the drug and therapeutics research that I'm going to talk about. In all of biology, a gene is made of DNA, and that lives in the nucleus of our cells. A gene is a recipe that tells the cell how to make a protein, and pretty much everything cool that our cells can do is done by proteins. So proteins make up the membranes of our cells, they make up the skeleton that holds the cell together, they are the control panels that switch on and off the genes, they make chemical reactions happen more quickly. Basically anything that happens in a cell is happening because a protein learned to do a very specific job. So each gene is a recipe for a protein, and the proteins are the machines that do things in our cell. In the case of Huntington's disease, we have the HD gene. We sometimes call it HTT, but it's basically the HD gene. And that's a recipe for a protein that we call Huntingtin, with an IN at the end of it. And when there's a small change in the sequence of letters in the gene, what's formed instead of the healthy Huntington protein is a protein that we call the mutant Huntington protein. Mutant is just from a Latin word that means changed, so it's an altered protein. But that mutant Huntington protein is harmful to cells. And we see evidence of that here in this very, very high-powered microscope picture of a blob of mutant Huntington protein. So this is the protein that comes from the abnormal HD gene, and what you can see here is it looks like a mountain because it's formed this huge blob or clump in the cell, and that is abnormal. So normally proteins shouldn't form these blobs. And down at the bottom of the mountain you can see these little things that look like sausages, and then even further down you can see there's little spherical mini blobs of Huntington. And so what that tells us is that the protein, the Huntington protein, has a tendency to be sticky, and when it sticks to itself it can also draw in other proteins away from their normal functions, and they get stuck in these blobs or aggregates. And that's one of many ways in which the Huntington protein messes around with the very delicate functioning of our cells. And our brain cells, which we call neurons, those are the cells that do all of the thinking and all of the cool stuff our brains can do. Those are particularly sensitive, fine-tuned machines. So when anything like this big, clumsy, sticky protein is floating around in those cells, bad things happen. And that's basically 100% for sure the cause of every case of Huntington's disease. So we have this certain knowledge that the HD gene is the cause of Huntington's disease, and that the mutant protein, mutant Huntingtin, is the thing that causes damage in cells. Biology is slightly more complicated than that, and because our DNA is so precious and it's essentially a family heirloom that's been handed down over generations, over thousands and hundreds of thousands of years, our cells are very good at looking after our DNA. They don't want that DNA to end up being damaged by anything that happens in the cell. So the DNA stays locked away in the nucleus of the cell. And when the cell wants to make a protein, it makes this carbon copy, like a photocopy, or like a snap that you might take on your cell phone, that basically it can take into the kitchen and make the recipe, follow the recipe to make the protein. So that is it's a sort of like a working copy of the gene. So it's a much smaller structure. It's made from this molecule called mRNA, which is basically like DNA, but it's a single strand and it's very slightly different chemically. So that mRNA is, we call it the messenger molecule, but you can also think of it as a sort of a photocopy of the recipe. And that mRNA message molecule is the thing that is repeatedly interpreted by the cell. It reads that message again and again and again. As long as that mRNA is there, the protein keeps being made. The cell will keep making protein according to the instructions in the mRNA. If the mRNA isn't there, the protein cannot get made and it doesn't get made. And that's where this idea of gene silencing comes from. We've been talking about gene silencing for 15 years now in Huntington's disease, but we're slightly moving away from that term because some people fear it gives the impression that we want to switch off the Huntington gene completely. We don't want to do that, and even if we did want to, it seems to be impossible using current technology. So instead what we really want is a volume control knob, not to silence the gene, but to turn down the volume slightly. So instead what we talk about nowadays is Huntingtin lowering, but we're talking about the same technologies when we use that phrase. And the leaders in the field of Huntingtin lowering in Huntington's disease for sure are Ionis Pharmaceuticals and their partner Roche, who have done almost all of the heavy lifting over the past 15 years to turn this idea of switching off the Huntington gene into a reality that has now been tested and continues to be tested in human patients. So what Ionis have pioneered is a new class of drugs. These are drug molecules that are made from DNA. So DNA is this double-stranded molecule, the famous double helix. What Ionis makes are drugs that consist of a single strand of DNA. It's chemically altered DNA so that it's a short single strand of DNA that has the properties of a drug. So it's turning the idea behind our genetic code into a way of making drugs. Those drugs are called antisense oligonucleotides, or ASOs. Antisense means it sticks to DNA. Oligonucleotide means it is itself a short stretch of DNA. But we usually call them ASOs. And so Ionis has this technology that they've been working on for decades. And in fact, they now have several licensed drugs that exist that have this ASO structure that are targeting the switching on and off of very specific genes targeting diseases, including a brain disease of children called spinal muscular atrophy. That was the first time that an antisense or ASO drug was licensed for a brain disease. It was a drug called Spinraza in SMA, and that drug is transforming the life of those children with that disease. Huntington's is a similar disease. It's a neurodegenerative disease, but we want to switch off a harmful protein, and that's what Ionis has been working on with lots and lots of partners over the years. So the idea is very simple. Basically, the drug molecule is designed with a sequence so that it will stick very precisely to the message molecule that comes from the Huntington gene, but it won't stick to the message molecule that comes from any other gene. So it's a very, very targeted approach that is designed in a computer and tested over many generations in a lab to make sure that the drug only targets the message molecule, the mRNA, from the Huntington gene. When the cell detects that something has stuck to its RNA, it has a whole mechanism of its own already built in to delete that abnormal pairing of mRNA with something else. And so the cell basically then deletes that message, it chews it up so that the mRNA isn't there. And as we've already seen, no mRNA, no protein. So the rate of manufacture of the protein is reduced. This doesn't get rid of any protein that's already been formed, but actually the cell is already trying to do that. The cell already has mechanisms for trying to chew up protein that's already been made. So the idea is that by lowering the production of mutant huntingtin, you give the cell a much better chance of being able to recover some of the damage that's been done, get rid of some of that protein that's built up, and then have less to worry about moving forward. So it's pretty cool. This is just one graph from 20 years of development of this drug. Basically, the vertical axis here is the amount of the message molecule, the mRNA, in neurons, and the horizontal axis is how much of the drug has been administered to those neurons. And essentially, if you give a bigger dose of drug, a bigger dose of ASO, you end up with less of the message molecule. And we see the same thing in terms of how much protein is there. So these are very targeted, effective drugs. And as I've said, they have some history of being given to humans. What about Huntington's disease? Well, the first time this kind of drug was tested in a mouse model of Huntington's disease was 2005. And it's a mouse model where the mice look okay when they're born, but they rapidly develop symptoms of Huntington's disease, or symptoms similar to Huntington's disease. The drug was first given to these mice after they'd already developed symptoms, and the hope had been maybe the mice would stay the same, or maybe they'd get worse but more slowly. What was actually seen when the drugs were treated with these kinds of Huntingtin lowering drugs was that the mice improved. They got better at controlling their arms and legs, they got better at thinking tests, and even the protein buildup that was seen on the brain under the microscope, that actually improved as well. So this was a real success in mice. What about humans? Well, there's a bit of a problem, which is that the mouse brain is about the size of your little fingernail. Human brain is much bigger. It's slightly smaller than the size of your head, actually. So human brains are very difficult to treat compared to a mouse brain. What is working in our favor, though, is that the human brain is surrounded by this fluid that we call cerebrospinal fluid. And this is a clear fluid that's produced by the brain that surrounds it and protects and nourishes the brain. It's not just in the head. The cerebrospinal fluid, or CSF, is in a continuous column from the brain right down to the tailbone, so it goes all the way down the spine. And what that gives us is basically a back door. This is it's not really blue, by the way. This is just so you can see where it is on the slide. So this is an MRI scan showing where the CSF is at the base of the spine. And you can see the spinal cord is the sort of thin grey strip that runs down the blue. So at the base of the spine, there's essentially a back door into the nervous system. You can easily stick a needle in there and collect samples of spinal fluid. And anyone who's had a lumbar puncture for the HD Clarity study will know that that is hopefully quite straightforward and not as bad as you thought it was going to be. Once the needle is in there, all you have to do is attach a syringe full of drug and squeeze, and the drug is administered into the CSF, from where it quickly spreads into the brain. And we know that these DNA-based ASO drugs can absorb into the brain, the substance of the brain, and get into neurons, which are the cells we really want to treat. If you took this drug as a pill, it would get digested by the stomach. But this method of delivery into the CSF, which we call intrathecal injection, or IT injection, is a really handy way of getting these kinds of drugs into the brain. As a consequence of this, which is a relatively new thing, I mean, this is not something that you expect to be doing when you first sign up to be a Huntington's disease researcher. I had to go back, and many other people like me across the world had to go back to training, and I was taught by our colleagues who treat blood cancers how to give these intrathecal or spinal injections. So this route is commonly used in spinal anesthetics. It's also used to treat certain forms of cancer where chemotherapy needs to be given into the nervous system. So for me and many other people, 2015 was a year of learning new skills, and I had to learn how to give these intrathecal injections. What we designed, along with Ionis and their partner Roche, was what's called a multiple ascending dose trial. And it was also a first-in-human trial. So it was for those of you who are pharmacology or drug development geeks, this was a phase 1b/2a multi-ascending dose first-in-human trial. So there were 46 patients involved in this trial. And at the start of the trial, very small doses were given alongside some patients receiving placebo doses. As the trial went on, successive patients were given either placebo or higher doses of the drug. And gradually, as the trial went on, we were able to increase the dose. And during that trial, no safety issues emerged that would tell us that we needed to stop increasing. So it was a multi-ascending dose trial. And the first two doses, or the first two injections, were given in September of 2015. On the left, the first injection was given in Vancouver by my friend and colleague Blair Levitt, and on the right, about 6 days later, I gave the first injection in Europe. We both looked quite relaxed, but actually this was a very stressful week for us, and probably rather stressful for our patients as well, who've given permission for us to show these photos of a really quite historic moment where the first dose of this drug was given to the first patients with Huntington's disease. Very brave volunteers who agreed to take part in this study. 18 months later, we were finally able to reveal what the trial had shown. No news is good news when it comes to drug development, and it takes a long time to run even a small trial like this. The results frankly knocked our socks off and were better than we had ever dared to hope. What you're seeing here is what happened throughout the course of the study at the highest and second highest dose. So the left-hand panel is 90 milligrams per dose, the right-hand panel is 120 milligrams per dose. And each person's CSF mutant Huntington concentration is on the vertical axis. So the higher the points are, the more mutant Huntington is still sloshing around in the nervous system. So what we want people to do, everyone is 0 at the start, and then the change is measured from that. So what we want it to go is as far down as possible during the course of the study. The grey lines are individual patient trajectories, but the green lines shows the average of what happened in each group. And what you can see very clearly is that in both groups, the average mutant huntingtin concentration gradually fell during the course of the study in patients who received just 4 doses of the intrathecal ASO huntingtin lowering drug. This was brilliant because not only were we able to show that the drug was safe and well tolerated, but we were also able to show that the drug was doing what it was supposed to do. It successfully lowered mutant huntingtin using the best measure of huntingtin we have for the level in the brain. And if you look across all data in all groups, what you see here is a slightly different graph, but the same basic concept. Lower is better, but each group from left to right is a different dose of the drug. And you can see that in the high doses, 90 and 120 milligrams, we achieved between 40 and 60% lowering in most of the patients. And we have a model of the relationship between protein level in the spinal fluid and the brain that tells us that that's very likely to be a kind of degree of lowering that would be expected to be beneficial if we continue doing it in patients. We don't know for sure, and that's a really, really important caveat. We don't know yet whether this drug will slow the progression of Huntington's disease. We get a possible clue from this analysis, and this is showing all of the data in all of the patients, including those who received placebo injections. The vertical scale here is a composite measure of clinical severity. What that means is basically you test people's movement and thinking skills, and then you squish all of those assessments into a single number, where the higher the number is, the better the patient is doing. And what we're looking at here is how much that number changes during the trial. So 0 means the patient didn't change at all during the trial. A score of 2 means that their clinical features improved by 2 points. Now you can see that the grey dots here are the placebo-treated patients, and some of them improved during the trial, which tells us that there was a big placebo effect, and that's something we need to bear in mind. Some of what we may be seeing here may not be placebo effect, but the horizontal axis here is how much the mutant huntingtin level changed. So if it fell by a lot, then those points are towards the left-hand side of this graph. And what you can see is that basically, and the statistics confirm this, on average, the patients who had bigger reductions in mutant huntingtin were also the patients whose clinical scores did better. The patients with the smallest huntingtin reductions, so those are in the bottom right-hand corner of this graph, and most of them are grey, which makes sense. Their huntingtin levels didn't fall because they didn't get the drug. But they, on average, deteriorated more than the patients who received the active drug. This is what's called a post-hoc analysis, so it was done after the results were in. What that means is that we cannot put too much confidence in it. We have to treat it with extreme caution. We definitely cannot look at this graph and conclude that the drug works to slow the progression of Huntington's disease. What this is a very tentative signal that it is the right thing to proceed with this program. We kind of knew that already though, because of the success that we had in just lowering the huntingtin concentration. But what this tells us is that we have the right to expect that successful huntingtin lowering, if it continues to be safe, is something that will produce improvement in the disease. But we do not know that yet. Okay, so to reiterate, we know that this drug lowers mutant huntingtin in the nervous system. We don't yet know whether it will be clinically effective. That's what we need to figure out. You will probably have seen some of these headlines. December the 11th, 2017, I was on holiday trying to do Christmas shopping in Budapest, but I spent the whole weekend on my mobile phone talking to lots of journalists. This was big news at the time. That was completely appropriate because this really was one of the biggest breakthroughs that we've seen in any neurodegenerative disease in recent memory. However, that victory was entirely achieved at the biochemical level. It was a victory of numbers and molecules. It is not yet a victory of changed lives. Hopefully that will come next. I had a patient that I saw in clinic shortly afterwards who is an actor, and so we were talking about films, and he was asking me what this result means for Huntington's disease. And for some reason this sequence from the movie Predator popped into my head. So you've hopefully seen the movie. It's an Arnold Schwarzenegger film in which a group of soldiers is dropped into the jungle and is being attacked by a very dangerous, invisible enemy, the Predator. No one's ever seen it before, and everyone who's ever encountered it has died which probably sounds quite familiar to people who are living with Huntington's disease. So it's a terrifying enemy. Halfway through the film, they see this creature out of the corner of their eye and they all fire their guns into the jungle, and it looks like the creature's escaped, but then they look down on a leaf and they see this fluorescent green liquid, which is it's clearly the blood of the enemy that they've been trying to kill. And Arnold Schwarzenegger looks at this blood and then he turns to the camera and says, "If it bleeds, we can kill it." And I think that's what we've achieved for Huntington's disease. With this success in the first Huntington lowering trial, what we have accomplished is not an end to Huntington's disease. This is still a problem for everyone who was ever born with the HD gene. But what we've shown for the first time is that this enemy, the mutant Huntington protein, which appeared to be unassailable, it does in fact have a weakness, which is that we can cut off its supply in a controlled and reversible way at its root and prevent or lower the production of this mutant protein. If we can do that, we have every right to expect that we will achieve clinical success. When Arnie says this to his colleagues, he doesn't know for sure that they're going to defeat the predator. Spoiler alert, they do. What he is doing is making an assertion based on new evidence that is accumulated from a solid encounter. And it's a reasonable belief at this point. I think that's probably where we are in the battle against Huntington's disease. So where we are now is that the drug, which used to be called HTTRX, has been taken over by Roche, and Roche's American arm, North American arm, is called Genentech. So all of their drug names begin with RG. They've given this drug the name RG6042. As far as I'm aware, that doesn't mean anything. It's just a number. But that's what the drug is currently called. Roche needs to run what's called a phase 3 clinical trial. The drug will be licensed if it shows that it can alter the progression of Huntington's disease in a phase 3 clinical trial. There's no way we can get the drug licensed on the basis of the evidence that we have at the moment. So we have to run this efficacy trial, sometimes called a pivotal trial. That trial has been announced and is open for recruitment as of now. The trial is called Generation HD1. It stands for something, but it's so complicated, it's not even worth talking you through what it stands for. This is going to involve about 660 people at sites across the world. So this is huge. This is the first time a Huntington lowering drug has ever been tested in a phase 3 trial in any disease. Huntington's disease, it's the first time any drug company's ever tried to use an ASO to lower the production of a harmful protein in a brain disease in a phase 3 trial. Generation HD1 is already up and running at one site in Canada, and there are 5 other sites. You can see them on the screen here. Edmonton, Vancouver, Halifax, North York, Ontario. Actually, there's two sites in Ontario and a site in Montreal. So 6 sites overall in Canada, which is pretty good considering how many sites across the world are enthusiastic about running this trial. The HSC and their website will be able to point you in the direction of how to get involved in this trial. There are, not to be a Debbie Downer, but there are a few things I need to say that, because it's a situation where there's a lot of excitement, which is justified, but we also need to proceed with caution and cautious optimism. So here's a few things that I want to say. First is, we don't know whether this drug works or not. If it works, that's great. It's possible that treating people in the long term will produce side effects or harmful effects of the drug that we didn't see in that short phase first in human trial. So this being involved in the Generation HD1 trial is not a guarantee that your Huntington's disease will be cured or anything like that. One person in 3 in the trial won't receive any drug for 2 years. But even if you're on the active drug, it's possible that your HD would be completely unaffected, or that it might even be accelerated, or you might encounter some other side effect that makes you feel worse. So this is not access to a treatment that we know works. What it is access to the opportunity to help the world find out whether this drug works. So the main reason for being in this study should not be because you yourself want to get better, although that would be great. It's not something that's on offer. And actually, even if the drug works really well, let's say the drug slows the progression of Huntington's disease by 80%, which would be phenomenal, way more than we have any right to expect. A year later, even if your Huntington's is chugging along at 20% of the current rate, you're still going to feel worse. We'd have to reverse the disease for people to feel better, and I think the chance of us seeing that is very, very low. So to be realistic, even if you manage to get into the trial and receive the drug at the high dose, and if the drug works really well, the outcome will still be you'll feel worse. You would be, if the drug is working well, you'll still be better than you would have been if you hadn't got the drug, if you see what I mean. But people mustn't go into this trial with the expectation that they'll suddenly start feeling better. This is a drug which is a long-term treatment to slow the progression of HD so that people end up feeling the same or approximately the same for as long as possible. The other thing to note is that this trial is going to be hard work, okay? 25 lumbar punctures. I don't know anyone in the world who's had 25 lumbar punctures before. So this is a very ambitious, intensive study. It's 2 years of your life, and each lumbar puncture and each intrathecal injection will mean that you have to come to hospital. You may need to stay overnight. You'd certainly need to stay all day. And in between times, there'd be assessments, blood tests, brain scans, urine tests, questions about your mood, questions about whether you're feeling like you want to harm yourself. It's a pretty heavy set of procedures, and it's 2 years, so it's a long time to be involved in such an intensive study. Obviously, there's an upside to that, which means that you're getting a lot of attention from experts in HD, but the downside is that being in this trial will sort of become part of your everyday life. It's really for people who are prepared to take that burden on themselves on behalf of the global HD community. It also requires people to be quite well supported. So really the people that were recruited into this trial will be people who have a very good family support network and people who can look after them and let the trial centre know if the person's not doing so well. So it's hard work. The other thing to note is that, like all studies, this trial has quite strict inclusion criteria and exclusion criteria. So these are very strict criteria on which we have to decide who's going to be able to be in the trial and who cannot be in the trial. You have to be able to walk, you have to be able to talk clearly, and you have to have fairly early Huntington's disease judged by your level of independence. But the one inclusion criterion that I think is going to cause a lot of conversations between patients and carers is this thing called the CAP score, CAP score. This stands for CAG age product, and it's basically a formula that looks at your CAG repeat count, the number of abnormal repeats in your Huntington gene, and it looks at how old you currently are. And it's a formula that basically is a measure of how long you've had too much mutant Huntington. And this score, it has the advantage that it can be calculated quite easily on the basis of things we generally know about patients already, the CAG count and the age. And it's well known that it predicts the rate of progression. So patients with a high CAP score will probably progress more quickly. That's bad news in general, but it's good news for a clinical trial. Because if you expect your patients to progress quickly, and if your drug works really well, that means you'll get a quick answer from the smallest number of people to whether the drug is working. We call this statistical power. It gives the study the power to answer the question it needs to answer as efficiently as possible. The downside is that this CAP score will make a lot of people ineligible to be in this trial, and that's really not negotiable because the numbers basically cannot be changed. The table that you can see here is something that I've worked out as a sort of ready reckoner to look at your CAG repeat count and how old you currently are, and basically for each CAG count, the number on the right shows how old you would have to be in order to be in the study. So if you have a very low abnormal count of between 36 and 39, by the time your CAP score is high enough to be in the trial, you'd already have exceeded the maximum age limit of 65. So people with those CAGs won't be able to be in the study. People with a low CAG of 40 or 41 some of them will be eligible, but if your CAG count is 40, you'd need to be at least 63 years of age at the time of screening in order to be in the study. With bigger CAG counts, it gets easier to be in the study. So if your CAG is 45, you only need to be 35 years of age or more. But very young people may struggle to be eligible for this study. People with much larger CAGs over 50 would basically be eligible at any age, except that the trial also has a minimum cutoff of 25 years. So if your CAG is 55 and you're 23 years of age, even if your CAP score meets the threshold, you won't be able to be in the trial because you also have to be over 25. So every trial has these criteria. This is a slightly unusual one, and I think it's worth talking about so that you may have some understanding of why we need to have this conversation about CAP score. It is something that we expect to make the trial more useful and more efficient. My advice, if you find that you are enthusiastic to be in the study but can't be, is see that as a good thing. What that means is that this drug will be tested on your behalf by other people. So that means you can sit back and relax while other people do all the hard work. The drug is going to get tested anyway. You being in this study as a particular person, clearly that may be a disappointment to you, but it doesn't mean that the drug won't get tested. And if the drug works, it will be made available for licensing as quickly as possible. And there will almost certainly be other opportunities during the course of this study for you to volunteer to take part in other drug trials and other research projects. So what about the outcomes? How could this trial and this trial program end? It seems to me that there are three possible outcomes. There's a brilliant outcome, there's a kind of good but not surprisingly awesome outcome, and then there's a crying cat emoji outcome, which is a disappointing result. I think the most likely outcome, from what I believe about this drug and what we know about how it works, is that the trial will run from start to finish as expected, and if we're lucky, we will see some positive effect slowing the progression of Huntington's disease. That would be the best news we've ever had in HD. But that news will take about 4 to 5 years before it reaches us, because that's how long it will take to recruit these 660 people, and then for them to all run through the study to completion, and then to analyze the results. So brace yourself for a long haul. It's going to be 4 or 5 years before we hear from this trial. Bad news can happen at any time, and that's the nature of clinical trials. There's a possibility that the trial will be ended early, and there's a possibility that if that happens, it will be because of an unexpectedly awesome result. That's something that it's nice to think about briefly, but we must not for a moment expect that. This trial has not been designed to finish early. No trial is designed to finish early. We need to expect that the trial will run to conclusion. Some trials finish early because some unexpected harm has been uncovered, and that's quite common in drug development. That doesn't mean that lowering Huntington is a bad idea, but it might mean that a pause is needed to figure out what's happening. So it's also possible that that this program could end early and in a disappointing way. If the trial goes well, you may hear some good news about that, but that doesn't mean the drug will be available instantly. So the drug has to be licensed by bodies like the FDA or the European Medicines Agency. There's often a second level of review by government bodies that decide on health spending. Insurance companies or nationalized healthcare systems have their own ways of deciding whether the drug is worth spending money on. So there may be a second battle to get that license. And in fact, in the UK, the NICE is locked in a battle with the drug company for that spinal muscular atrophy drug that I mentioned earlier. So that drug's still not available for prescribing in the UK because of these administrative hurdles. Hopefully that will be quick for Huntington's disease, because it's clearly a situation where there's huge need for good drugs, but we don't know for sure. The other thing to say is that even in the event of a phenomenal result or a really good result, this is still a drug that will need to be given by regular intrathecal injection, spinal injection. So it's a bit like when diabetes was discovered as being caused by insulin deficiency. That's great, we have treatment for diabetes now. Oh, but you need to give yourself an injection every day, and a lot of people can't do that. Some people won't be able to cope with lumbar punctures every month, every 2 months, or however often we can give it. Maybe we can make those injections easier to give, but this is still going to be a drug that's somewhat difficult to get into the brain where it needs to be. So this new future that we're creating may be a future of lumbar punctures to prevent the onset of Huntington's disease in people who actually feel fine. That's a big burden for healthcare systems. We're worrying about all of that stuff. Hopefully it will be sorted, but I just don't want to create the impression that a rosy future is just around the corner. This is all going to be hard work for everyone. It's a different kind of hard work, and it's going to be a better future. But it's important to stay grounded in reality. So what if the trial doesn't work? What if the drug doesn't work? What if something goes wrong? What if some side effect emerges? That's possible, and we've seen that happen in other programs. I think if that happens, we'll figure it out. We'll figure out why it's happening. We'll figure out what can be done to make that less of a problem in future. Maybe with this drug, maybe altering the dose, the frequency, the mode of delivery. Maybe there's some second treatment that can be given to treat the side effects of the first treatment. Maybe we have to try a new drug that tries to lower Huntington by a different approach. If that happens, lowering the cause of the disease is still a good idea, and we still have that victory from December 2017 to fall back on. We still prove that the predator monster can be made to bleed, and we still have this sort of save point. We don't need to go all the way back to 1993 to figure out that we can lower mutant Huntington. We probably just need to figure out a different way of doing it. And that brings us on to the fact that this is not a one-horse race, not by any means. You hear a lot about Roche and Ionis because their program is certainly the furthest ahead, and it's the only one that's now enrolling in a pivotal study. But there are probably about a dozen different organizations and companies that are trying to lower mutant Huntington in Huntington's disease by various mechanisms. So Ionis and Roche is one. But Wave Life Sciences also has 2 ASO drugs that they are testing in human patients, including Canadian patients, and those I'll talk about in a second, but that's another ASO. Unicure and Voyager are 2 examples of companies that are attempting to set up trials of gene therapy-based Huntington-lowering drugs. I'll talk about that as well. And further ahead down the road, a company called Shire has had its zinc finger protein drug taken over by a Japanese company called Takeda, and that is a drug that acts directly on the DNA to reduce the production of the mutant Huntington protein. You can also see PTC Therapeutics in there. They're shooting for the moon. They're trying to develop a drug that doesn't have to be given by spinal injection, hopefully a pill that you could take regularly that will lower the production of mutant Huntington and no need for any spinal injections. So you can see clearly that this is a very wide field, and lots of clever people are trying to attack this problem from multiple angles. We have, as the drug companies and sports people say, multiple shots on goal. A quick word about the Wave Life Sciences program. As I've said, this is another ASO drug program, so it's a very similar program to the Ionis Roche program, but this is trying to do something slightly different. What Wave are trying to do is just lower the production of the mutant protein without lowering, or with much less lowering, of the healthy version of the protein, which is also produced. In almost all cases, people with HD have inherited one healthy copy and one harmful copy of the Huntington gene. In this case, it was mum that passed down the harmful copy to this person. That harmful copy is a recipe for the mutant Huntington protein, but the healthy copy is a recipe for the healthy Huntington protein. And we don't know for sure what happens if you lower both copies of that gene, but if you could lower only one, it seems like that might be beneficial. In other words, you might be able to produce bigger amounts of lowering without any of the theoretical risks that come with lowering the healthy protein. As I say, those risks are theoretical at the moment because we don't yet know whether lowering both proteins, which is what's happening in the Ionis Roche program, whether that has any adverse events or adverse effects that are due to lowering the wild type protein. We call it the wild type protein. It's the healthy protein. It's not named after me. So what we're trying to do is, what Wave is trying to do is to target the abnormal or mutant copy of the protein without lowering the healthy copy of the protein. That program, using 2 different drugs, relies on some very delicate genetic sequencing techniques to identify people that the drug would work in, because it targets little tiny changes in the DNA that are sort of time-traveling passengers that have been traveling through time with the Huntington mutation. So there's little changes in the DNA that are silent in themselves, but they tend to travel alongside the mutation as passengers. And the drug targets those regions that contain those changes, and that's how it's able to target the mutant but not the normal copy of the gene. As I say, that drug is currently in a trial that's similar to the trial that finished in 2017 for the Ionis Roche ASO drug. We haven't heard any news yet, which is, as we've said, no news is good news. That's the WAVE program. Thinking ahead, we heard some news in the past couple of weeks that was quite significant for the next generation of therapies. So these ASO drugs have to be given regularly, and currently the Ionis, Roche and WAVE drugs are all being given monthly, although in the phase 3 trial it'll be every 2 months. But what if we could give a single drug dose once in a person's life, and that would then treat them for the rest of their life? I don't know what a cure is, to be honest. It means different things to different people, but certainly something that you only have to do once and never have to do again is closer to a cure than something that has to be given every month. The Ionis Roche program will tell us whether what the long-term effects are, and it has the advantage that if any adverse effects emerge, if we stop the drug, we can remove those effects. So a single treatment that lasted a very long time would have the advantage of being long-lasting, but the disadvantage that any side effects that emerged could also last a long time. What I'm talking about is this next generation of treatments called gene therapies. This is basically either changing the DNA in the brain or introducing new genes, new DNA into the brain that then become a part of that person's brain, and they live with that altered DNA in the brain for the rest of their life. The picture that I'm showing is an indication that these treatments currently have to be given by a neurosurgical operation. So it's not quite as shown here, but a hole is drilled into the head and a small needle is passed down, and the drug is then injected directly into the substance of the brain. So these are kind of a hardcore next level of invasiveness with the potential advantage of producing very long-term lowering of mutant huntingtin production without having to have repeated treatments. The way they work is very similar to the ASOs, except that there's a couple more steps involved. So what's injected is a virus particle. This shown here is a hexagon, a little virus called AAV, which has had all of its own DNA scooped out of it and has had a completely synthetic genome injected into it. That DNA gets injected into the brain, and the virus infects the brain cells, and in the process it injects the DNA into the brain cell, where it becomes incorporated into the genome. It becomes part of the DNA that then lives inside that neuron for the rest of its life. That gene ends up being copied in much the same way as any other gene is copied, as we talked about at the beginning. So what you end up with is a new stretch of mRNA that comes from the artificial gene that was put in the brain by the virus. That mRNA is basically then going to do the same as the ASO drug that we talked about. So that sticks to the mRNA message from the Huntington gene. And the same thing happens as before. When the cell detects the mRNA stuck to the drug mRNA, it deletes it. So we're achieving the same thing. We're still targeting that mRNA message molecule, but what you can see here is that although the level of protein is reduced, the gene that makes the mRNA stays in the brain, and so that mRNA drug is produced again and again and again, and hopefully for the lifetime of the patient. So a single treatment should produce sustained lowering of mutant huntingtin, and that could really help us reach a new level of what we could achieve in terms of treating Huntington's disease. So where we are now is that every trial that's been run is involving patients who already have some symptoms of Huntington's disease, usually early HD, because those are the people who are best able to tell us whether drugs are working. What we'll certainly see as these trials proceed is whether we can slow the progression, whether we can then treat later stages of Huntington's disease. But the real breakthrough, I think, will be whether we can prevent the onset of HD. Can we treat people early when they're young adults, or even when in childhood, in order to delay or prevent entirely the onset of Huntington's disease? It's possible that we could do that through drugs that have to be given regularly, but that's a goal that's probably going to be easy to achieve if we can find ways of delivering one-off gene therapy treatments to the brain. The announcement that came out a couple of weeks ago was that Unicure, which is a Dutch-American gene therapy company, has just received FDA approval, not to prescribe the drug, that's many years away, but FDA approval to begin human trials. It's called an IND, or Investigational New Drug Approval. So the Unicure program is now the most advanced gene therapy Huntingtin lowering program for Huntington's disease, and we expect to be hearing more during the course of 2019 about exactly what Unicure's proposed clinical trial design is going to look like. I think we'll also probably hear about FDA approvals for other gene therapy approaches from other companies like Voyager, but that remains to be seen. So all of this is great, and clearly it's a time of huge progress. We've talked about the potential downside and the need for patience and cautious optimism. Preventing the onset of Huntington's disease comes with its own problems. So this is my friend Jeff, who at the time of this photograph was 40 years old. And Jeff carries the HD mutation but doesn't yet have so-called manifest HD. But if we don't do something about it, he will get HD at some point. If we treat Jeff now with a drug that works, even if it works completely, because he looks okay now, in 5 years he'll still look okay. So that's great, but it's also a bit of a problem. If he looks fine in 5 years, is that because the drug is working, or was he going to look fine anyway, just because the random chance of HD is sometimes like that? What we need is ways of measuring whether the drug is working. We already have shown you one of those in the form of mutant huntingtin. So measuring the mutant huntingtin level in CSF tells us whether the drug is hitting its target. It may not be able to tell us whether the drug is saving brain cells. So my team at UCL has been working on this biomarker, this measurement in spinal fluid CSF and also in blood, which will hopefully be one of several measures that we'll be able to apply in the clinic and in these prevention trials of HD to help us figure out whether the drug is helping neurons or doing nothing or even harming neurons. So we're better equipped now to be running these sorts of trials, and this kind of breakthrough in biomarkers that really helps us run trials has come about because people have volunteered for studies like HD Clarity. The work of CHDI Foundation has been absolutely pivotal in running these studies. Enroll HD is another really good example of a study that's enabled us to really leapfrog other neurodegenerative diseases because we have this big knowledge database and group of patients and families that we know are standing up to try and help us contribute towards this fight. The Critical Path Institute is a spin-off from the FDA and is basically a non-profit foundation that is working with us now to develop these biomarkers, and hopefully will help us to get the FDA and the other regulatory agencies to agree that we need to be running prevention trials in Huntington's disease and figure out how to do that. And so in the future, I think what we may be heading towards is a situation where people can have a genetic test for HD at any age, but instead of just being information that helps them plan their future, what it becomes is a trigger that helps us give them treatments. And those first treatments, or the treatments that are given earliest in life, may be the sorts of pill-based treatments that I was talking about that might have some small benefit but can be given fairly easily over a long period. We may then be looking at blood tests or lumbar punctures to look at the spinal fluid as a means of measuring in those people who look and feel completely fine whether the drug is working, whether it's slowing the disease, whether the disease is progressing more rapidly than we would like, and then we will have, I think, multiple options for escalating to more invasive treatments, be that intrathecal spinal injections or injections of gene therapy drug into the brain. And eventually, I think we will be challenging the genetic fate of people who were born in HD families. Another Arnold Schwarzenegger film, Terminator 2, contains a memorable sequence in which Sarah Connor, the mother of John Connor, who's going to save the Earth, inscribes into a table. She vandalizes a picnic table, but it's after the apocalypse, so there's a lot less concern about picnic tables. Anyway, she scratches "no fate" into the table, and the full phrase that she's quoting is, "The future's not set. There's no fate but what we make." This has always been true in Huntington's disease, and it's always been the case that if more people volunteer to be scientists, if more people volunteer to be participants in HD research, if more people fundraise, if more people donate, if more people help their relatives to take part in research, if people raise awareness, together we can change the fate for the next generation of people in HD families. Everything that I've presented has been made possible thanks to people who have volunteered their own time to take part in research or to help other people take part in research. They are a global army of HD research heroes, and I salute every one of them. We are together, I think, in the best position we've ever been in for changing the future. It's a hard road ahead with lots of hard work for everyone, whether they're in these trials or not. But I believe that we can achieve a completely different future for people affected by Huntington's disease. Thank you for listening to this presentation, and I look forward to seeing you again in the near future, and thank you to the Huntington Society of Canada.
Thank you so much to Dr. Michael Hayden and to Dr. Ed Wild for sharing all of this important information. I feel so inspired, and I know Ralph, looking down on us, feels the same way. I hope you've enjoyed today's Community Education Forum. They say it takes a village to raise a child. Well, I feel like we've created a village where everyone looks after each other and supports each other. The future is bright, and my heart is full of hope. I truly believe that Ralph's dream of a world free from Huntington's will be realized.